Hello, everybody. It's Mr. Cadell. I hope you're doing well. I'm doing well. I laugh because it's early December. Oh my gosh. Seems like everything's on fire right now. Anyway, let's get through this together, and I'm hopeful. I'm absolutely hopeful for the future. Let's talk about bacteria and archaea, chapter 27. There's just a small amount of this chapter that we need to do. It's part of this unit that's kind of a hodgepodgey unit. We have chapter 26 with phylogenetic trees. Here we are with bacteria and archaea, and then the next three chapters will be on plants. And they'll just be little bits of the important information that exists in those chapters as far as the AP people are concerned and the DE class across the street. This is kind of the last time that we will actually go through something that's super small. After this, it's all big picture. It's plants, it's a little bit on body systems, and then ecology, and then we just go bigger and bigger and bigger. So last bit with the cells, so get through it with me. There's definitely some good stuff in here. I would love to do this entire chapter. I think a lot of people would admit that bacteria are awesome, and they're way cooler than eukaryotic cells in many ways. Of course, eukaryotic cells are much more complicated and complex and allow for greater ability, like to break down food efficiently and all these things, more genetic material to be stored. But anyway, we're just going to do 27.2 is first up there, and then a little bit of 27.1. But I just want you to think about bacteria for a second. First of all, there are more bacteria in a handful of soil than there are people that have ever roamed this planet. Over 15 billion. If you can wrap your head around that, that's a lot of bacteria. The oceans are also a bacterial field. Another place that there are a lot of bacteria is your skin. In fact, on a square inch of your skin, I would like you to look at the back of your hand right now. Square inch, 32 million bacteria. That's just mind-boggling, those numbers. The question becomes, are bacteria good for us or bad for us? And I'm going to argue that obviously they're both. We have tons of bacteria that live in our digestive tract. In fact, I've heard an estimate to be around 1 billion for an adult human. These things help our immune system. They help us produce vitamins. They help us with help us digest food. They just help do a lot of things. And then there are others who argue that we aren't really even here at all. And I've made that argument to you before, that in fact, you're just basically waves of energy, but the little small amount that's here are these atoms which have come from space, of course, and you can condense them down into the head of a pin. And then some would argue that we have 10 times more bacteria on us and in us than we actually have eukaryotic human cells, whatever those are. So you're 10 times more of a bacterium than you are human. And then if you take all the genetic material, because the cells are a lot smaller, they're like 1,000 times smaller than a eukaryotic cell on average. Some people say because there's so much more genetic material with the bacteria than the eukaryotic cells, that you're only 1% human as far as genetically here. So if you take that small head of a pin amount of mass, only 1/10 of that is human. It's not, or 1% of that is human. Aren't you feeling insignificant? Don't you just love nature and biology? I hope that you do. Again, what we're seeing here is what we're going to see for the rest of the textbook. And what this is a picture that I took from the College Board. So I have a picture of our textbook's cover, and you can see that there. And then what I want to highlight here is the highlighting. And who did that was the College Board. And they said, okay, these are the themes that we're going to ask you to teach your students, have students understand, and then we're going to test them on these concepts on the AP exam. So the point is 27.3 and on, even though there's a few interesting things that I'm going to have you look at, they're not as important. I want you to trust me and trust Lauren too.
Okay, then Lauren's going to catch you. Okay, it's called the trust fall. Okay, trust fall. Ready, set, go.
All right, so I promise I won't leave you out in the cold. Make sure that you take a look here before you jump into this chapter and any future chapter, because there's no way we can get through like 30 chapters in the second semester of this book. We even have half the time that we did in the fall. So the point is that we're just going to like jump around, and that's fine, but you just have to understand I'm trying to communicate with you what you need to know and do. So look here first, for sure. Well, these are the two of the few learning targets that you guys need to be able to do by the end of this chapter. And I see terms here like number 2, cell walls. And then down there in 3, I see something with gram-positive and negative bacteria. And you can see that up here on the board. And I'll go over that in just a second, what that is. So the title of concept 27.1 is Structural and Functional Adaptations Contribute to Prokaryotic Success. There are many who believe that prokaryotes are the most successful life form on this planet. If you have to imagine that they've been around for billions and billions of years. And I'm going to show you some reasons why they've had so much success. And there's way more species of them than there are of anything that's eukaryotic cells. Just like the nervous system or neurology is the study of these nerves up in the brain and spinal cord, is the kind of the least understood physiologic concept amongst physiologists. For sure, amongst biologists, the least amount is known about bacteria. We don't even know most of the species that exist. We don't really know all their little complexities and nuances, and we're learning new stuff all the time. Well, the cell wall is super important, and that's something that's different. Certainly plant cells have cell walls, but bacterial cells do have cell walls, and it gives them an advantage over most eukaryotic cells, although fungi and plants both have cell walls. They help maintain the cell shape. They provide this protection from the environment. There's always these terrible environments that these many bacteria live in. And they prevent the cell from bursting in a hypotonic environment where they're going to take in water because they have more tonicity than the surrounding solution. And you can imagine that they'd be turgid or filled up, but they're not going to burst. The cell wall does many things for them. Now, cell walls are mostly made up of these things, peptidoglycan monomers, and you can see the picture of it there. And what you see there are some carbohydrates, and then if you look at the bottom, you see some peptides. These are peptide bonds between amino acids, so it's called peptidoglycan, like for glucose or some sort of sugar. So it's kind of a sugar protein mix thing. And these things come together. These are a network of sugar polymers cross-linked by polypeptides. And there was this Danish scientist in the 1800s, and his last name was Graham, and so we call this thing the Graham stain, which is what we can do in order to try to classify bacteria, because his stain, so I have it right here, and I really enjoy doing this in class as well and showing there's great movies on bacteria, and specifically antibiotic resistance, because that is a big issue to add to the pile of issues that we have today in our world. But we'll talk about that a little bit next time. But we're going to Graham stain some of your bacteria at the end of this talk. Hopefully in class, we'll do it when I see you. So we're going to try to classify. So the idea is hey, if we get a bunch of bacteria, and what microbiologists, like I used to have a student whose mom worked at the Vail Valley Medical Center, and she was a bacteriologist or a microbiologist, and she would take, have you ever had like someone swab the back of your throat or something at the doctor's office, and they put it on a little petri dish, a culture plate, and then over a couple of days, you can imagine that's pretty awesome to look at after a couple of days, but it will grow up this bacterial colony. And what this person would do is they would take some of those bacteria and they want to identify it. They want to say, what is this? So then we can use the right medicine to treat it and help out the patient. So this is the first test that they always do, because you can split bacteria into two very distinct categories here. Well, here's what they look like once they've been stained. If you notice, the stain I held up was pink. It was purple. And these are gram-positive bacteria, which hold on to the purple stain. I have a little movie showing you how this is done, which I'll show you in a minute. The peptidoglycan traps the crystal violet stain. Bush League. It's like a clown show here. You can see that peptidoglycan layer way up there at the top of the
program to illustrate how the Gram stain procedure is able to distinguish gram-positive and gram-negative bacteria by representing the staining events at the ultrastructural level. This particular animation is one of two in this series, showing the staining of gram-positive bacteria. with critical structures of the bacterial surface represented schematically. The circle at the lower right tracks how the bacteria would appear in the microscope if they were examined during each step of the staining procedure. Prior to staining, the bacteria would be transparent and invisible in the microscope. After fixing the slide, it is first flooded with crystal violet for 1 minute and then washed. The stain colors the bacterial cell wall blue, and the bacteria will appear blue in the microscope if examined at this point in the procedure. Next, the slide is flooded with iodine solution for 1 minute and then washed again. During this step, the iodine and crystal violet combine to form a larger complex within the layers of the cell wall. Microscopically, the bacteria will appear dark blue or black after this step. The slide is now rinsed with a decolorizing agent and acetone alcohol solution. However, the crystal violet iodine complexes are not washed out of the thick and tortuous layers of the gram-positive cell wall, and the organisms remain dark blue in color. Finally, this slide is counterstained with neutral red or safranin for 1 minute and then washed again. The red stain also confers color to the bacteria. However, the red color is not apparent because of the persistent dark blue stain that dominates the microscopic appearance of the bacteria.
I know it's not that exciting, but we are going to do it because it's like I said, it's a common procedure done in identification of bacteria. There's another type of bacteria that you might find after you do a gram stain, and these are gram-negative bacteria, and I hope you don't find many of these. And today, when we do this in class, make sure that your friend doesn't have many of these, and if they do, stay away from them. Reason why is because gram-negative bacteria are the bad bacteria. And what has happened here is that they have less peptidoglycan. You can see that peptidoglycan layer, but it's not on the outside any longer. It's actually, if you look at the left side of this picture, it's actually underneath a phospholipid bilayer. It's on top of one, and then it's underneath another. And the idea is that the purple stain isn't able to work as well, and so they look a little bit more red. They look a little bit more red or pink under the microscope. But why this has ramifications for people is that our immune defense doesn't work as well trying to knock down these bacteria as our immune defenses and white blood cells can knock down gram-positive bacteria a little bit better because it's just more straightforward. There's a peptidoglycan layer there. We're evolved to identify that. This can also be toxic for a few reasons that are mentioned in your book, and they're more likely to be antibiotic resistance because they just have this extra phospholipid bilayer there. See there in the picture that there's an outer membrane, a peptidoglycan layer in between, and then a plasma membrane underneath. Well, I thought I was going to show you a movie on that, but again, relax, I'm a professional. There's some weird things about cells, bacterial cells. Just remember this. They have a circular chromosome that exists in a nucleoid. It's not a nucleus. It's just kind of an area of the cytosol, but it's unprotected. It's just there. There's no nucleus that's keeping it safe. And then once the mRNA is made from that, there are floating ribosomes that are a little bit different. You'll read in your textbook than the ribosomes that exist in eukaryotic cells, and then they translate that message into protein. So that happens. Also, you can see there's a lot of these little nuances, which are the third and fourth page of the chapter. There's fimbriae, which are these little hairs. They've got flagella that help with motility or moving. We'll talk about the sex pilus in a moment. They have capsules, they have endospheres. These are just things that they've evolved to have to keep them kind of protected in the environment. They can last a long time outside of an organism. And then they can maybe be in the soil or something and then get into organisms after years of just sitting there. It's pretty crazy. It's some of the adaptations that prokaryotes have. Well, let's pause and most importantly, shut me up. Go ahead and press pause and speak out loud your answers to these two learning targets, please. Well, where we're going next is 27.2. And here is the, I guess the title of 27.2. Rapid reproduction, mutations, and genetic recombinations promote diversity in prokaryotes. So they are the most diverse cell type on the planet, and here are the three major reasons why. Remember, they have tons of genetic variation, and they can evolve rapidly, mostly because they're short generation times. They can divide about once every 20 minutes. I want you to think about human reproduction. We create a new baby not only after we've reached puberty, and then that takes 9 months, and then you have to grow the baby up and make sure it's living on its own. That takes, in some cases, never happens, but decades for most kids to be able to get a job and do their thing on their. And so the point is that the reason why prokaryotes have been so successful, one of the reasons, is because they have quick generation times. This is called binary fission. It's very similar to mitosis for sure, but of course it's a little bit different. The offspring and cells are generally identical, but there's always mutations that have occurred due to outside influences or when the DNA was replicated. And actually, they use that to their advantage. It's all subconscious. They don't do it consciously. In fact, I read that every day in our gut, we produce 9 million E. coli bacteria. Those are the bacteria that help us digest food in the large intestine. 9 million bacteria that have never been created before. These are mutants. Now, some of them are going to have an advantage in that environment, and some won't have an advantage, and so they'll be naturally selected against. But the point is that if you create enough of these, this variation in any environment, natural selection will favor those that have characteristics that help themselves better than the others, and then those might move on, and we might get a new species of bacteria as a result of that. This allows for rapid evolution for sure. Well, I have a handout that has really 4 good pictures on it, in my opinion. And one of the pictures is this that's on the screen here. Go here. I hope that you have a hold of this handout. This goes over the 3 types of genetic exchange in bacteria: conjugation, transduction, and transformation. So I'll quickly go over these and then we'll be done. Additional diversity arises from genetic recombination. You can see here that prokaryotic cells can take up and incorporate DNA from the surrounding environment in a process called transformation. This often happens when a cell dies. You can see the one on the left there, and often the cell wall and membrane break open and the genetic material just goes out in the environment, and some other organisms, cells might take that up. Now, we try to use this every year when we do our P-Glow experiment. When you make the DNA glow, they're just used to getting DNA from the environment, so we can put those P-Glow genes out in the environment. They're uptaken often by bacteria and incorporated into plasmids, which are these small circular pieces of DNA. We've seen this before also with Griffith's transformation experiment. I think that the heat-killed cells here spit DNA out into the environment, and then the living R cells I'm getting it all wrong. But the point is that I'll just throw something. So the point is that some bacteria uptook some DNA that was out in the environment.
Some bacteria possess surface proteins that can transport DNA from closely related cells, allowing a process called transformation. When a bacterial cell dies, it can break open and release DNA, which can be taken in by closely related species and incorporated into their genomes.
Pretty sweet. You'll have a chance to reflect on all this in a second. Here's another type. This is called transduction. Transduction is the movement of genes between bacteria by bacteriophages. These guys accidentally, when they inject their DNA into a host cell, bacteria to try to infect that, bacteria to then have it make more viruses. Sometimes they will actually take up some DNA from that bacteria on accident, probably in these little plasmids, and then they'll just go and they'll infect another bacteria. And the point is then they're going to inject some of the DNA that was in that original bacteria that they were infecting into another bacteria. So it's just called transduction.
Transduction. In this process, bacterial DNA is transferred from one bacterium to another inside a virus that infects bacteria. These viruses are called bacteriophages or phage. When a phage infects a bacteria, it essentially takes over the bacteria's genetic processes to produce more phage. During this process, bacterial DNA may inadvertently be incorporated into the new phage DNA. Upon bacterial death and lysis, or breaking apart, these new phage go on to infect other bacteria. This brings along genes from the previously infected bacteria.
Sweet, there's only one left. Here is a sex pilus. And what is sex after all? It's just the exchanging of, my hands away. It's just the exchanging of DNA and genes in order to make a novel new organism that's never been there before. And so sometimes through conjugation, these guys are going to conjugate here with this tube essentially, and it's going to allow cells to connect and pull together for DNA transfer. And then it's a process where genetic material is transferred between cells with this little tube. It's just something that bacterial cells have evolved in order to transfer DNA between each other. Go ahead and turn to your neighbor or to your favorite stuffed animal and make sure that you speak these out loud. Go ahead and press pause. Thanks. If we were in class, we would do a gram positive and negative stain right now, but we're done with the lecture. Thanks for listening.